Okay, perfect. Yeah, it's my pleasure to introduce Ryan Canfield. Ryan has recently finished a PhD at University of Washington, working alongside Dr. Amy Osborne and Greg Horowitz, and his research focuses on brain-machine interfaces, neural plasticity, and neural encoding and decoding. Well, with that, Ryan, the floor is yours, and we're going to listen to your talk. Thank you.
Great. Yeah. Thanks for inviting me and being here. I'm gonna, as you know, be talking about our work investigating the structure of neural activity and motor cortex during reaching. And we've made a few changes from the preprint that you have seen based on responses to reviewer comments. So these weren't major changes, but some of the figures and explanations have changed in ways that we hope are a little clearer. Can you see this laser pointer? Yeah.
Yeah,
Yeah.
you can see that.
So yeah, as you know, brain-computer interfaces are a promising approach for the millions of people with motor disorders. They record neural activity from the brain, decode movement, and send control signals to an actuator, like a cursor on a screen or a prosthetic limb. But they can only produce the correct movements if there is task information in the recorded neural activity. And so for this reason, BCIs use our general understanding and knowledge of the structure of information in the brain to target implants that record neural activity. Many approaches use somatic topic maps to target implants to cortical areas where electrical stimulation evokes movement in the hand or arm. And then most clinical studies use fMRI guided implants while users imagine hand or arm movements. And in this case, grasping, where the red and yellow areas represent the activated parts of cortex, and these squares are the implants. Um, but the spatial resolution of these targeting approaches is on the order of multiple millimeters. And this has been fine, and this resolution has been fine because commonly used implants, microelectrode arrays like Utah arrays that are shown here in this figure on the right, are kind of a similar scale. And they also have rigidly defined geometrical constraints, both across the cortical surface and in depth. And so it's difficult to take advantage of a more nuanced understanding of structure in frontal motor cortex with this technology. Yet new and emerging technology is enabling us to record specific neural populations and maybe even neurons. We're seeing that th thin, high density laminar probes like neuropixels and ultra flexible thread-like electrodes are being developed. But the spatiotemporal structure of single neuron and population activity in frontal motor cortices has not been well characterized. And this lack of understanding limits our ability to leverage these new technologies for brain-computer interfaces. In frontal motor cortex, we've seen multimodal evidence suggesting that task information is heterogeneously distributed across these areas. Electrical stimulation studies that show the cortical areas where electrical stimulation evokes different movements are intermixed. For example, the areas where stimulation activates shoulder movements are intermixed with areas where stimulation activates wrist movements. Um, Anatomical studies also suggest that cortical projections in motor cortex don't terminate with a uniform spatial distribution, and multi-neuron resolution mapping studies like microelectrocorticography or microECOG show that some patches of cortex seem to have activity that is more predictive of movement than other patches of cortex. And this is shown here with data from a micro-ECOG mapping study where surface potentials were recorded from motor cortex while a monkey performed a reaching task. The black line represents the arcuate sulcus, and each pixel is an electrode contact. And we can see that the predictive activity tends to be most clustered in these two areas. Yet most single unit recordings pool neurons from across frontal motor cortices, and they're commonly conducted with either acute single electrode recordings, shown on the left, where each dot represents a single electrode penetration, with the gray and black just being different monkeys, or with chronic microelectrode arrays implanted into the hand-arm region of motor cortex, shown on the right, where each square represents a microelectrode array implant. And since analyses of data collected with these approaches tend to pool all recorded neurons, it kind of assumes or implies a homogeneity in neural responses during movement. And so we wanted to ask how task information is spatially distributed at the neuron level. So to address this, I use NeuroPixel probes, which I know you guys are mostly familiar with, but they're high density laminar microelectrode arrays that allow me to sample neurons in depth with high spatial resolution. And I recorded from two monkeys, each with chambers over motor cortex that encompass the putative hand and arm region. We used a modular implant where we replaced the natural dura with a silicone artificial dura, shown here in blue, and the neural pixel probes could easily penetrate this artificial dura. And then I attached a grid on top of this that allowed me to track the spatial position of probe penetrations. So this allowed me to record from many spatially precise locations across the surface of motor cortex in each monkey, with each black dot representing a recording site, left being rostral, right as caudal. And since each penetration was with a laminar probe, I was able to track the three-dimensional position or coordinate of each recorded neuron. And collectively, I recorded over 1,000 units from each monkey. Um, and so while recording, we had the monkeys just perform the standard delayed center out reaching task, where the monkey's hand position was mapped to control the position of a cursor on a screen. Monkey initiated the trial by moving the cursor to the center target and then had to hold there until go cue, well, one of the 8 peripheral targets appeared. At go cue, they had to move the cursor to the target, and if acquired successfully, they were rewarded. So for the upcoming analysis, I focused on just reaching related activity, so the activity around movement onset in this reaching phase of the task. And this task was well learned. You can see the trajectories of each target are very similar. And then we further quantified this behavior similarity across days by comparing the trajectories of each day with every other day by computing the pairwise correlation. And we can see the behavior across recording days for each monkey is stable. And I had to zoom in on the color bar to make any differences visible. So this consistent behavior combined with recordings across a large three-dimensional volume allowed me to use a pseudo-population approach for further analyses. I compared how much the activity of different pseudo-populations modulated with reach direction by applying a cross-validated linear discriminant analysis, or LDA classifier, to predict target direction based on neural activity. Target direction is just one form of task information, but when I say task information going forward, I'm only referring to target direction information. And this is also the decoding approach used for all of the decoding in this project. So we quantified the heterogeneity of task information across all recorded units with single unit decoding accuracy, which tells us how well each individual unit predicts target direction. And we see a non-normal distribution that was skewed right compared to the chance distribution in gray, which was constructed by just shuffling target labels. And so from this, we see task information seem to be unevenly distributed across the recorded units, with this right tail of units having much higher task information than others. And these patterns were not explained by proxies for unit sort quality like SNR or true spike percentage, defined by defined by refractory period violations. So then I investigated if there was some spatial organization of task information across the recorded neurons that led to this observed heterogeneity. So I first grouped units into populations based on the depth that they were recorded from, kind of regardless of cortical surface location. I computed the decoding accuracy for each population while controlling for the number of units, and plotted the accuracy against depth with these error bars representing the 95% confidence intervals. All of the confidence intervals overlap, suggesting that no one depth consistently encoded more task information than others. I then regrouped units based on the surface location that they were recorded from and computed decoding accuracy for each of these populations while again controlling for the number of units and plotted the accuracy on a spatial map of the that shows the recording site location on the cortical surface. Again, this black line represents the arcuate sulcus. And now we see differences where some areas have higher accuracy and some have worse. And the error bars on the right represent the 95% confidence intervals, matching the same color on the spatial map on the left. So we find that decoding accuracy was significantly different for the highest and lowest accuracy sites, and these results were also not related for proxies for recording quality like kilosort drift, or differences in firing rate or total unit count. And so this suggests meaningful differences and that task information is heterogeneously distributed across the cortical surface. And the areas with high task information can be separated by many millimeters from each other and have lower task information sites between them. So next, I wanted to investigate the temporal structure of these populations. So we've seen that single unit responses are heterogeneous during movement. Each plot here represents the activity of a different unit, with the red and green traces being the average activity to different targets. And the gray traces are the X and Y velocity. And they all have different response characteristics that happen at different times before the movement or are multiphasic or even not really responsive. But recent population approaches that pool neurons from across motor cortex and look at the collective activity patterns in a population space have been useful to reveal structure. And so I applied these tools to look at how the temporal structure changes across spatially distributed neural populations. So for this comparison, I used canonical correlation analysis, or CCA, and this allows me to compare the dynamics of two distinct neural populations. So it starts by taking two separate populations. Here now, they're each are composed of units recorded at different locations on the cortical surface. Compute their individual principal components, or PCs, and then project the neural activity patterns onto the top 10 PCs. And the further results were not sensitive to the number of PCs chosen. So here these plots now represent or show a low-dimensional representation of how the neural activity patterns evolve in time, or their temporal dynamics, each color being the average activity on reaches to a different target, starting at movement onset. Then I used CCA to compare the structure by aligning the temporal dynamics to find the directions that maximize the correlation between the low dimensional trajectories of the different populations. And we get a correlation value that represents the similarity of the dynamics, with mode 1 being the direction that the dynamics are the most similar in, mode 2 being the direction the dynamics are next most similar in, and so on. When we apply this analysis to compare the dynamics of neural populations from each pair of recording sites, we see that the dynamic similarity of different neural populations varies across the cortical surface. And also just another note that the CCA values we're reporting here are lower than some other published literature because we're controlling for population size. So we then asked what best explains this variability in dynamics similarity. Our first thought was that similar dynamics are driven by spatial proximity. We hypothesized that site pairs that were physically closer together would have more similar dynamics because nearby neurons are more likely to be anatomically connected. Instead, I found no discernible pattern or relationship between the CCA correlation of two neural populations and the physical distance between them across the cortical surface. So spatial proximity did not clearly predict similar dynamics. Next, I hypothesized that similar dynamics follow a direction or axes across the motor cortical surface. I focused on the rostral caudal axis because of known anatomical and functional gradients in this direction. And so here I expected that caudal sites would have the most similar dynamics because caudal motor areas tend to be most associated with movement execution. So I organized sites by their position on the rostral caudal axis and compared the dynamics to neighboring populations as defined by their position, the rostral caudal position. And now a relationship seemed to emerge for monkey one. Consistent with the hypothesis, caudal populations tended to have more similar dynamics to each other than rostral populations did to other rostral populations. But for monkey two, the relationship is less clear. So rostral caudal position had an inconsistent relationship to similar dynamics across monkeys. And another note, this is kind of robust to the specific axis choice that or the specific, yeah, axis definition. So next, I hypothesized that task information drove similar dynamics, where recording sites with low task information would have different dynamics, but recording sites with high task information would have similar dynamics. And this hypothesis is supported by past observations of the heterogeneous functional maps from multi-neuron mapping studies that I mentioned earlier, and that anatomical cortical-cortical connections can span multiple millimeters, providing the architecture to support spatially distributed coordination. So I sorted populations now by their task information, and we see a strong relationship to dynamic similarity. And so from evaluating these three hypotheses, we see that task information best predicts which neural populations have similar dynamics. So now this suggests that spatially disparate neural populations with high task information have similar neural activity patterns. But I wanted to probe this further by asking if the relationship between task information and similar dynamics was at the site level or unit level. So on one hand, coordinated dynamics could occur with insights regardless of task information, or coordinated dynamics could occur regardless of the location and instead are based on task participation. So I return to single unit decoding accuracy for all of the recorded units, and we see that some have a lot of task information and some have very little. So I first split units into two groups based on their decoding accuracy, low and high. And for comparison, I also had random neural populations with randomly selected units. Then, as an internal control, I arbitrarily split each low, high, and random population into two groups. So I now have two neural populations, each with units that have little task information, two other populations, each with units that have high task information, and also two populations with distinct randomly selected units. And I can now compare the dynamics of each population with high task information to another population with high task information, as well as other populations with little task information. And so from these, I found that the high-high comparison had the most similar dynamics compared to the low task information units or randomly selected units. The black line and shaded gray region represents a lower bound computed from intertrial interval data. I'm only showing monkey one here, but the results are consistent across monkeys. Then, as a test of how this compares to site level dynamics, I repeated the random unit selection, but restricted units to those that were recorded at the same site. And the dynamic similarity of these populations is still lower than the high task information populations. When I plot the CCA correlation across the top 10 modes with the error bars representing the 95% confidence intervals, we see that these patterns persist, suggesting that they are not just driven by spurious correlations. The high task information populations have the most similar patterns of neural activity and suggest that similar dynamics are driven at the unit level. So then, kind of on the other hand, we asked if we're just observing a trivial relationship between task information and population dynamics. Conceptually, task information and population dynamics can be independent. And this is a schematic kind of depicting the different options with the different colors representing different task conditions. So you can have correlated dynamics with little task information. The neural activity patterns of each population are similar, but the conditions are not separable. You can have correlated dynamics with high task information. The neural activity patterns between populations are similar and each condition is separable. Or you can have uncorrelated dynamics with high task information, where the neural activity patterns of each condition are separable, but they're not correlated. And so we empirically tested this by forming random pairs of populations and comparing their CCA correlation to the average decoding accuracy of the two populations. So now each gray dot represents a pair of randomly selected populations. We see a weak correlation between population dynamic similarity and, or sorry, between decoding accuracy and CCA correlation. And then we also compared it to the high task information subpopulations that I talked about earlier, and these seem to be outliers. So we further refined our analysis by only looking at the random populations that were, I guess, accuracy matched to the high task information populations. They were the ones that were in the same average decoding accuracy range as the high task information populations. So the x-axis is zoomed in quite a bit here. And even after this, we still see that the dynamic similarity of these populations are all lower than the task information populations. We can compare this distribution now to the ones described earlier as this ACT match distribution. And we see that the dynamic similarity of the high task information population is always the highest, suggesting that similar dynamics are driven at the unit level and not just because of a trivial relationship to task information. We then performed an additional control to test if our pseudo-population analysis approach was obscuring shared dynamics that were not task related. So we repeated CCA, our CCA analysis for the recordings that had enough simultaneously recorded units to split them into the low, low, high, high groups I described earlier. And we see that at least for the sites that we could test, our analysis was not missing obvious task irrelevant shared signals. And the gray lines now are connecting the comparisons from the same simultaneously recorded units. So the high task information populations consistently had higher dynamic similarity even during simultaneous recordings. And then to connect this back to spatial structure, I asked how the high task information populations were distributed and plotted their location on the cortical surface. And we find that they are spatially distributed across motor cortex and can be separated by 10 millimeters, suggesting that these neurons may be coordinating across large distances during reaching. And then lastly, we wanted to ask if these high task information populations all had similar task representations. Could a decoder trained on one population generalize to another population? So to test this, we turned to align decoding through CCA, where two populations can be, we can align two populations with CCA by projecting their activity onto the, sorry, by using CCA to find kind of the shared axis and projecting the activity of each population onto this shared axis. Then we can train a decoder on the activity of one and test it on the activity of the other. So we can repeat this for all the low and high population comparisons. And we find that this high task information population that is spatially distributed has similar task representations. So kind of in summary, we've seen that task information and dynamics are heterogeneously distributed across the motor cortical surface, with subsets of neurons seeming to tightly coordinate during well-learned movements. And we think this is consistent with ideas of functionally defined networks. Um, but also kind of leaves open questions about how it relates to how other movement variables that motor cortex encodes. But it also, our results also highlight that accounting for spatiotemporal heterogeneity will likely be important for improving BCI performance. It highlights the need for improved mapping techniques to target implants based on task information or to design decoders that account for the different dynamics of different neural populations. And so with that, I'd like to thank everyone in the Orsborn lab, both past and present, for their help and support, especially Bea and Tomo, who helped me collect the data and set up the analysis pipeline. Thank you to the vets and staff at the Primate Center and also our funding sources. Yeah, and that's all. Any questions? I'll go back to this slide.
Hey, thanks, Ryan. Let me see if there is any question from people. Yeah, Armin.
Yeah, I had a little bit of a technical detailed question. So, so do you use all the reaching execution time for your decoding analysis, or do you use a specific time point, or do you run the decoder at each time point and get the time point that is the highest accuracy for that specific single unit? Which one of these are you?
We found the time that I use the time that the cursor leaves the center target as movement as to define movement onset because that's I know that the cursor is moving at that time, and then I used a window starting 50 milliseconds before that to 300 milliseconds or to 250 milliseconds after that. So a
Right,
set window for every trial. And then it's LDA, so I average the activity for each trial to get one number.
right. Okay, thank you.
Yep.
Thanks. Yeah, I also have one question about the laminal results that you showed earlier in the talk. You mentioned that there is no difference in the overall amount of decodability, but did you have a chance to look at whether the onset of decodability or onset of information arriving also changes when you look at different depths?
Not in a rigorous way. I early on I looked at that a little bit, but didn't find anything super obvious. And I think a lot of that is just because I would expect that to be related to layers and motor cortex has such complicated kind of cycle topography that I'm not convinced that I think we were rarely recording perpendicular to layers. And so it's kind of hard to estimate, um, yeah, th you know, things like that. Um, yeah, that's my thought. And then yeah,
Yeah,
does that
so
answer the question?
yeah, so you're saying it would be hard to ensure that you're perpendicular to the layer, so and then hence
Yeah,
analyze
I didn't
any.
see anything. I guess, yeah, in summary, I didn't see anything from my initial analysis,
Yeah.
but I don't think that's because there isn't anything there. I think that's because we didn't have the data to show that.
Thank you.
Yeah, I have a question, Ryan. So I have a problem with I have a conceptual problem. So you have these site locations that have low task information. Um, so let's say those are like locations that are just driven by noise. There's a really low reliability of the activity within that location. Wouldn't then it be trivial that the CC element of those locations would be low to the other locations?
You talking about like if it's not like if it's just all noise and not task responsive?
Yeah.
Yeah, we this was actually a point that our reviewers brought up, and so we repeated our analysis by restricting our results to just task responsive units. Um, And so we separated them into task responsive, non-task responsive units. And then we were able to replicate our analysis, like the main findings of the CCA findings even though we were restricting our analysis to task responsive units.
How do you define task? So it's just that the task you define task responsibility based on decodability, right? Of the
No, decodability is like the separation of activity to different targets.
just
Uh,
activation to the movement. Okay.
yeah, so task response, yeah, exactly. So this task responsiveness is a significant modulation compared to a baseline window during our movement onset period.
Okay. Okay. Thank you.
Hmm.
Jorn, you could go ahead.
Yeah, I mean, related to this, it sounds like you got a good amount of grief from the reviewers on this point, on exactly how trivial this is. Is that correct? Or
Um, I think it was things we were thinking of. So it didn't seem like none of the major points were about these, but we felt like the reviewers brought up kind of slightly tangential points that made us think that we needed to nail down some of these points a little bit further, and
yeah,
that's why we added more.
but I just wonder what you think, or what's your interpretation conceptually here. Um, So I'm just trying to get my head around this, what you think this means. So I appreciate that you could have, I mean, the alignment is just based on the temporal dynamics, and that could be, you could have a region that has consistent temporal dynamics but doesn't encode for reaching direction at all, or a region that encodes for reaching direction, or two regions that both encode for reaching directions but don't have any common temporal dynamics and don't align in their dynamics. So I theoretically you could construct them, but like why? Okay, so you don't find that. What is it? How do you interpret this? I mean, what's the deeper meaning of this? I mean, this is
Like,
to some degree, is this surprising to you?
so I think there's a couple points to respond to this. I think first of all, we wanted to show that there are differences in dynamics depending on the populations that you choose, and we haven't seen that much in the literature. Second, yeah, sorry, you seem puzzled.
Yeah, I'm just wondering, like, how should I think about this paper? So from a high
Yeah,
level, I mean, the statistical
well,
issues, yeah, I mean, they're tricky, obviously, and it is true that if you're in a region that doesn't have any consistent temporal dynamics and doesn't encode for movement direction, it won't align with any other region, right? I mean, it's just that's just, if it's noise, then noise doesn't correlate, right? So and doesn't align. Uh, so that's kind of trivial in that bookmark end, and you saying that, well, we excluded those, but so what's the deeper point here? So that basically is your conclusion that most of the stuff that does encode for directions also has very similar dynamics,
yeah.
or are you stressing the fact that the dynamics might be slightly different in rostral than in caudal areas?
No, we're not. We're saying that it seems more like like this. If you're encoding task information, you seem to do it in similar ways across the cortex. That doesn't have to be the case. And
Across the cortex that you measured.
yes, of
Yeah,
course.
so
I mean, that's always the
right.
caveat with
So
all of
we're talking more
neuroscience,
than two
right?
premortem. Yeah. So I mean, do you think this would look different in SMA, like prefrontal
Oh,
cortex?
absolutely.
Likely, right? Yeah.
Yeah, but that's
Yeah.
why
Yeah.
this paper is more motivated from the translational perspective of brain like that's why I spent so much time
Oh, I see. Okay.
motivating it from the
Okay.
translational perspective of brain computer interfaces
Mm-hmm.
and like the need to think about these differences when you're designing your entire closed loop system.
Yeah. I mean, because you started with somatotropy, and this is a little bit slightly, it leads away from your actual study because you only studied reaching direction, right? And one
In this paper, yeah.
in this paper. And so one obvious concern is that, well, if they, like, given that these neurons probably are tuned for other bodily movements, finger movements,
Absolutely. Eye movements,
and so on. So now with
reward.
BCI, that's of course tricky, right? So
Yeah.
now you have these mixed selectivities, and you can't assess them here because you only have arm movement data. So what do
We
you?
actually
Yeah.
we don't, yeah. So in this paper, we're just showing this obviously, but I mean, we have eye movement data and we have some projects in the lab trying to look at how that eye movement data interacts with the hand movement data. The different grad students working on that.
Hmm.
Um,
Yeah, more relevant is probably wrist and finger and so on. So they're all going to be tightly interdigitated, right? And so you could have you can kind of think about all kind of different types of codes that
yeah,
things can code for, right? I mean, they could code for, especially when you go more rostral for the spatial outcome, right? And it doesn't matter whether you do wrist, a little bit wrist or not, whether the wrist is fixed or not, and so on and so on. So there's all kinds of complicated games that you could do with
yeah. I mean,
mapping, right?
that's the complication with all of motor cortex
Hmm.
research, right? I mean,
Okay, good. Thank you.
Yeah, I also have okay, Andrew, go ahead.
Yeah, I just wanted to nail down a little bit more. Oh, sorry, my camera's off here. I just want to nail a little bit more down about how you envision changing decoder design based on this result.
yeah. I don't think I have a super clear idea, but I think it depends on the application. I mean, so some of, the work out of the Batista lab suggests that the decoder you choose and how that relates to the dynamics impacts the ability to learn and performance. And so I think if I remember correctly, there's a recent paper that Emily Obi published that it's really hard to change temporal dynamics. So maybe if you want a generalizable decoder, you would need to sample from different populations with different dynamics to be able to find that. That's all kind of just off the cuff right now.
But so is the insight with respect to the decoding algorithm or from
Yeah.
which parts of
Well,
rostral caudal cortex you're recording from, or some
I mean,
combination
I
of both of
think
them?
I think like so I guess there's kind of 2 points, points I see mixed into this question. And first is that um it's like you're talking about targeting implants, and I think that can be done in two ways, either with actually targeting Utah array or neural pixel inserts, or it can be done as recording technology advances. It suggests we're going to be able to record more and more neurons, and then doing some sort of feature selection online. And so the decoding algorithm would probably involve, in my mind, some sort of feature selection and some sort of, decoder design in the mapping and how that adapts and with all of the, adaptive algorithms and stuff. Does that help?
Yeah, I guess I'm just having a hard time. If there's no the simple, I guess I'm not seeing the overall message to some degree, but the simple-minded thing is, if you have a decoder that is at all sophisticated in its design, the stuff that doesn't carry information relative to the outcome variable, to the degree which you need to kill it, I think
Yeah,
is
but that assumes that some of Amy's other work suggests that the decoder design itself impacts how the brain changes and learns. And so I think that it would probably, thinking about, and then from what I was saying about the Batista paper, the dynamics also affect how the brain can learn decoders. And so this is a there's complicated interactions in that area.
I see. So yeah, I think that's a good point about, like, if it's in a closed loop, if the decoder
Right.
is in a closed loop, how that might affect this. I'll have to think about that. Thanks.
Yeah.
Yeah, Ryan, I think I may be able to.
After you, I can ask another one.
Yeah, Ali, go ahead. Yeah.
I can go ahead. Okay, sure. Thank you, Ryan, for the talk. I'm Ali. Yeah, I had a question about the hypothesis 3 point, but maybe from another angle. I was thinking, like, maybe it goes back to like the details of electrophysiology, which I'm not fully aware of. But I was thinking under a super homogeneous hypothesis, if you had a cortical surface where all those locations were actually very, very homogeneous in terms of information representation, but then you poke with your, for example, neuropixel, and because you get unlucky, you don't get like as many informative neurons about the reaching directions for could there be other different reasons? That's the first question. And under these assumptions and hypotheses, then you would still get a flat line where lower decodable lower information neurons will give you less CCA correlation, and very high correlation neurons will give you higher correlations. So yeah, maybe the question is, could you get unlucky, for example, when you poke an area and get bad neurons? And then would that affect this analysis that lower decodable information is causing less similar dynamics? Because even if you have like a very high information group of neurons and you find their dynamics, and then you have a very low information group of neurons and their dynamics, I would think that if you want to align them and correlate them, you would still get a low correlation. Just because the data over there in the low decodable part of the data is just not good because of various reasons.
Okay, so you can there's definitely sampling variability every time you insert a neuropixels probe, but I think that our results suggest that so in your hypo, I'm trying to okay in your I guess scenario in this scenario the low information um, populations have different dynamics. So I don't think the sampling variability would affect that because we see the different dynamics even within a site. Is that what you're asking? I'm not sure I followed exactly what you're asking.
Maybe let me ask it like this. If you like poke an area which has a very high information, and then if you poke another area which has very high information, you could possibly find low correlation between their dynamics. Is that correct?
We're not finding that.
Yeah, you're not finding that, but I mean, like, just hypothetically, that would be possible. Like that you have two high information area with different dynamics, and then they don't match because they're from two different regions. Something that Jorn also mentioned, I think.
Yeah. I mean, like, so one way, so you're asking, like, you're like, my interpretation now is that you're asking if you can have task information with different dynamics.
That's a possibility, I think, that could happen. Now, my question is more like, now you have the same area with high information, and then you have this same area, you have this another area with now low information, and that low information is not is coming from, for example, an unlucky penetration in the cortex. And now your correlation is going to be definitely lower just because your data is not capturing those dynamics anymore. Could that happen, for example, when you use NeuroPixel in those regions? That
I think yes, but our results seem to suggest that it's not dependent on just like the specific penetration, but more on the units that you're recording.
Yeah, I mean, in each penetration, could you get like bad neurons and good neurons, and then when you only correlate the bad neurons with each other, you will get lower correlation, and vice versa? Is
That's
that
what I mean, that's what we're seeing. We don't know how much, like, we don't fully know how much, there's no ground truth to know how much of that is due to like inherent electrophysiology variability versus, and like being unlucky versus some properties of cortex. But all of our analyses suggest that it's more dependent on like where you're sampling rather than being lucky or unlucky.
Mhm.
I think I'm not sure if I'm answering your question.
Yeah, no, thank you.
Thanks, Ron. One quick question. I was just wondering if you're running both of these analyses, the CCA and decoding, on that 10 low dimensional PCA that you run as a
Decoding is just on firing rates.
pure firing rate.
Well, I mean, it's, like I mentioned, and in the answer to the first question, it's the average activity. Yeah.
So and also the CCA is also happening in full dimensional data without any like PCA in the pre-processing.
No, CCA is happening on the latent dynamics.
Yeah, so I was just wondering if this result changes if you just run everything, both the coding and also the CCA on the like full dimension data.
We haven't seen that, but I haven't fully done that control, but I have talking to everyone I've talked to has not seen differences in the results when they've done the analysis on latent dynamics or firing rates.
Yeah, thank you very much.
So yeah, I should probably do that at some point, but the reviewers didn't ask for it, and
Sure.
I'm not sure I have time.
That makes sense. Okay, thank you very much. If there is no more questions, we can just wrap up. Yeah, thank you very much again.
Thanks, Ryan.
Yep.
Thank you.
Yeah, have a good day.